All right, hello everyone. We will be covering chapter 10 today. The rest of the semester, we're going to be working on different areas of the nervous system. So we're going to kind of switch topics from muscular system over to the nervous system. So that'll be the focus of the rest of the semester. So as always, roots, prefixes, and suffixes. Most of these you probably will not know, with the exception of like by means to, uni down at the bottom means one. So as always, we'll go through some of these as we work through this lecture. All right, so nervous system, what does it do? It's our master regulatory system. It oversees all of our bodily functions. It receives information from the inside of our body and outside world, and it senses that information. It detects any changes that's going on. It looks at our blood sugar level, our thyroid level. What is our red blood cell count, white blood cell count? What's the temperature going on? It detects those changes, and then it sends that information to the spinal cord and that brain, and our brain and spinal cord integrates and processes that information, and it makes a decision as to whether or not something needs to happen. We talked about those negative feedback loops and those positive feedback loops. And if something needs to be done, then it will provide some sort of motor output to stimulate either a muscle or a gland, to provide that response. And again, everything goes back to maintain homeostasis. And we know that the brain is a center for thought, learning, memory, higher functioning, emotional processing, visual awareness. And when we look at nervous tissue, we can kind of break it up into two categories. Neurons, or nerve cells or nerves. Those are the types of cells that we really think of when we think of, the nervous system. Those are going to be what conducts those nerve impulses, those electrical impulses, and releases those neurotransmitters. And that's what's going to be responsible for detecting those changes from the inside world or inside of our body and the outside world. And that's what the neurons are responsible for, responding to those stimuli. So we have sensory neurons and we have motor neurons, but we also have neuroglia. Neuroglia is basically all of the supporting structures of the nervous system. They protect the nervous system. They support, nourish, insulate all of the neurons. They don't actually conduct those electrical impulses, but they're like the extracellular matrix of the nervous system. They're going to fill the gaps of the nervous system. So this is kind of like a picture version of these three general functions of the nervous system right here. So we have these receptors all over our body that detect what's going on. We have thermoreceptors that will detect heat. We have chemoreceptors that detect changes in chemicals like your blood sugar level. You taste chemicals, you smell chemicals. So all of your sensory receptors will pick up what's going on inside your body or what's going on outside of your body, and it will send that information to the brain and spinal cord where it will get processed. And remember, our negative feedback loops get processed up in the hypothalamus. And if something needs to happen, then the brain and spinal cord will send signals through the motor neurons and provide motor output to what's called an effector. And an effector is either going to be a skeletal muscle, a muscle or a gland. And we'll talk a little bit more about that later on. All right, we can divide the nervous system up into two different portions, central nervous system and peripheral nervous system. So central nervous system just consists of the brain and spinal cord. Peripheral nervous system is pretty much everything else. It connects the central nervous system to your other body parts. We have cranial nerves that lie near the brain and they originate off of the brain. They are not part of the brain tissue, so they're considered the peripheral nervous system. We have spinal nerves that originate off the spinal cord, but they are not part of the spinal cord, so they are considered the peripheral nervous system. And for now, we have two types of nerves. We actually have more, but for right now, we'll just say that we have two types of nerves, sensory nerves and motor nerves that we've talked about. Sensory nerves, also called afferent nerves, those go into the central nervous system. Motor nerves or efferent nerves go out of the central nervous system. So they kind of make sense. You touch a hot stove and those sensory nerves pick up your thermoreceptors in your skin, pick up the temperature of the hot stove, and those sensory nerves carry that information up into your spinal cord and into your brain. Your hypothalamus processes that information that the stove is hot. So your sensory nerves go into your central nervous system, and then your hypothalamus says, yes, that the stove is hot. I need to do something about it. And then your motor nerves that go out of the central nervous system is what actually removes your hand from the hot stove. And then we have this definition called ganglia. Ganglia is just a cluster of neuronal cell bodies that are located in the peripheral nervous system. And we'll come back to that definition later on. All right, so sensory nerves or afferent nerves. Those are sensory receptors that detect changes in and outside of the body. Typically, they have receptors on them. Like I mentioned before, we have thermoreceptors that detect heat. We have chemoreceptors that detect chemicals. Chemicals are like your blood sugar. You taste chemicals, you smell chemicals. We have baroreceptors that will detect changes in pressure. So like your blood pressure, for example. And these receptors actually convert that information into electrical impulses that will then get conducted through the nerves up into your central nervous system for integration or for processing, typically in the hypothalamus if we're talking about a negative feedback loop. Or a positive feedback loop. Then we have the motor component or the efferent pathway for the peripheral nervous system. And those are, like I said, neurons that transmit impulses from the central nervous system back down to that effector. And remember, effectors are a muscle or a gland. And the example here is eye movement. Or it could be your blood sugar level is too high. So your chemoreceptors would detect that your blood sugar level is too high, and then the motor component or your efferent pathway would stimulate your pancreas to dump out some insulin to lower your blood sugar. And the way that I remember sensory, afferent, motor, efferent is just the acronym SAME, SAME, sensory, afferent, motor, efferent. All right, picture version of this. Brain and spinal cord, central nervous system. Everything else, peripheral nervous system. We have sensory receptors that are part of your afferent division. And for our motor division, that will go to skeletal muscle, smooth muscle, cardiac muscle, glands. So any type of muscle tissue or gland is considered an effector. That's what your efferent pathway will stimulate. And we'll talk here in a couple slides about the autonomic and the somatic nervous system. So remember, brain and spinal cord is your central nervous system. Everything else is peripheral nervous system. Cranial nerves and spinal nerves are not part of the central nervous system. And we'll actually talk about cranial nerves and spinal nerves later on. All right, so we do have two divisions of the peripheral nervous system. The somatic nervous system and the autonomic nervous system. The somatic nervous system is part of the nervous system that we have control over. So think of skeletal muscles, your voluntary portion of the nervous system. So somatic nervous system is things that we have control over, skeletal muscles. Your autonomic nervous system is dealing with things that we don't have control over. So your involuntary portion, your smooth muscle, we don't have any control over our digestive system. Cardiac muscle, we don't have any control over how fast or how slow our heart beats. We don't have any control over our glands, how much insulin our pancreas dumps out, how much oxytocin our pituitary gland dumps out, how much norepinephrine our adrenal medulla dumps out. So that would fall under our autonomic nervous system. And within our autonomic nervous system, and I'll say I typically refer to the autonomic nervous system as the automatic nervous system because it happens subconsciously. We don't know that this is happening because it's involuntary or it happens at an unconscious or subconscious level. So within the autonomic nervous system, we have what's called the sympathetic and parasympathetic nervous system. You might know it as the sympathetic is fight or flight, and the parasympathetic as rest or digest. So here's kind of like a picture version of the breakdown of it. So we have the central nervous system as the brain and spinal cord, and it says interneurons. We'll talk about interneurons later on. Peripheral nervous system is everything else. We have those sensory, afferent, and motor efferent neurons. We can break up the peripheral nervous system into the autonomic and the somatic nervous system. The somatic is our voluntary, which goes to our skeletal muscles. The autonomic is our automatic portion. It's involuntary. And that efferent pathway will provide output to smooth muscles, cardiac muscles, glands. And we can break the autonomic nervous system down into the sympathetic and parasympathetic nervous system. Remember, sympathetic is fight or flight, and the parasympathetic is rest or digest. And we'll get into this adrenergic, noradrenaline, acetylcholine, cholinergic later on. All right, so organize the following list of functions according to the branch of the nervous system that controls each one. So heart rate. So basically we're looking at somatic versus autonomic. Which one do we have control over? Which one do we not have control over? So heart rate. That would be autonomic because we don't have any control over it. Walking, that would be somatic because we have control over it. Looking to the left, that would be somatic. Digestion is autonomic. Vasoconstriction, so that's where your veins or your blood vessels tighten up or constrict. That's autonomic. And picking up a glass, we have control over that, so that would be somatic. All right. A neuron in the aorta detects oxygen concentration in the blood and relays this information to the brain. This neuron is a blank fiber in the blank nervous system. So if it's detecting something and it's going into the brain, so we know that that would be an afferent neuron because it's going into the brain. So we know that we can cross off C and D. Now, is it autonomic or is it somatic? Do we have any control over how much oxygen content is in the blood? Or do we have any control over the aorta detecting the oxygen content in the blood? No. So we would cross off B because remember, somatic is our voluntary portion. Autonomic is our involuntary portion. So we're going to go with A. All right, so neurons actually vary in size and shape. We have many structural features on the neuron. One common feature of neurons is actually several common features, is that we have these things called dendrites. Dendrites are what receives the information. Basically, these are extensions of the cytoplasm. Remember, cytoplasm is that gel-like material that's within the kind of like the cell. So dendrites are just cytoplasmic extensions. And so these actually have tiny little receptors on them that would receive information. So this neuron has several different dendrites. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18 dendrites. But in reality, it could have 1,000. So these dendrites receive information. And a neuron could have, like I said, thousands of different dendrites. Then we have what's called a cell body. And that's this thing right here, cell body. We also, some older textbooks might call it a soma or a perikaryon. Rarely will you hear that. Inside this cell body, you'll see the nucleus, the cytoplasm, various organelles. We have these, if you remember back from like chapter, 2, 3 or something like that. We had the myofilaments and the myofibrils and that scaffolding to kind of keep the structure of the cell. Well, now we have these things called neurofilaments and neurofibrils. Basically, that's the scaffolding of the cell body. There's just cytoplasmic structures that help support the cell body and the axon. And then we also have these things called, that we haven't talked about, this chromatophilic substance, also called Nissl bodies. And I'll just say right now that these, it's just really unique to neurons. We only find these chromatophilic substances in neurons. Basically, it's a bunch of RNA, protein, and some rough endoplasmic reticulum. If you remember, rough endoplasmic reticulum helps with protein, packaging proteins, forming proteins, and basophilic material. Basophils is a type of white blood cells. And then also we find a bunch of organelles, mitochondria, produce ATP, lysosomes, Golgi apparatus to form vesicles, and some microtubules. So that's our cell body. And then we have this thing called an axon, and that's what this thing is. And that's what transmits that electrical impulse down through the axon, and it releases neurotransmitters and to either another neuron or some sort of effector. And remember, that effector could be, actually I have a little mistake here, which the effector could be a muscle or a gland. We'll just do that, a muscle or a gland. There we go. And we saw this last chapter. We actually kind of looked at this area right here, that axon terminal and the synaptic cleft, and it dumped out acetylcholine to stimulate that skeletal muscle. So a neuron can only have one axon, but it can actually branch off into many different collaterals. And last chapter, we saw that axon branched off into 4 collaterals. This axon branches off into 2 main collaterals. And then down here, it branches off into 3 separate collaterals. And then down here, 2 more collaterals. Then it branches off and branches off and branches off. So the axon can only, or one neuron can only have one main axon, but that axon can branch off. And we have this area here called that axon hillock. And that's where this electrical impulse is actually going to start. And we'll talk about the electrical impulse later on and how that starts to kind of create. And we'll talk specifics about numbers and stuff like that. All right, so dendrites, cell body, and axon. So when we talk about the pathway of the electrical impulse, or how information flows down the axon, it's always, always, always a one-way street. The dendrites receive information, then it will go through the cell body, then it will go down the axon. Never will it go in the opposite direction. It always goes dendrites, cell body, axon. And then let's say that we had another nerve down here. It would get picked up by the dendrites. Then it would go through the cell body, and then it would go down to the axon. Then it would go to the dendrites of another nerve, cell body, axon, dendrites, cell body, axon. So it's a one-way street only. All right, some other structural features of the axon. I already mentioned that axon hillock. That's this cone-shaped area that's right here. That's what comes off of the cell body, and that's how the axon arises. Remember that we can only have one main axon, or one axon coming off of that cell body. However, we can have many collaterals. So collaterals are those branches of the axon. So here we have some collaterals. Here we have some collaterals. Here we have some collaterals. Here we have some collaterals, many collaterals. So lots of collaterals. We saw last chapter that axon terminal. So axon terminal are those special endings right here. We also saw that synaptic knob. Those are the endings or the ends of the axon. So those synaptic knobs, and that's what's going to release those neurotransmitters. All right, Schwann cells. Now, we'll talk about later on the difference between central nervous system and peripheral nervous system, but Schwann cells are very specific to the peripheral nervous system. They're part of that neuroglia. Remember that neuroglia is the supporting structure. So Schwann cells are part of the neuroglia of the peripheral nervous system that wraps around some of the axon. And they produce this thing called myelin. And myelin basically is just a bunch of layers of lipids and proteins, so fats and proteins that wrap around the axon. And because it's fat-based lipids, it's going to be lipid soluble. So because it's lipid soluble, it prevents water-soluble substances from crossing that membrane, such as ions. And as this myelin gets wrapped around some of those axons, it creates this thing called a myelin sheath. So it's just layers and layers and layers of myelin. So it's this wrapped coating around some of this axon, composed of layers of Schwann cell membranes and myelin. And it acts as an electrical insulator, which I'll talk about here in a few minutes. And then we have these things called nodes of Ranvier, which are gaps in myelin sheaths or between Schwann cells. So if we go back up to this picture, here is myelin or myelin sheath. Here's another myelin or a myelin sheath. And that gap right here is a node of Ranvier. That gap right here is a node of Ranvier. That gap right here is a node of Ranvier. So myelin sheath, myelin sheath, myelin sheath, myelin sheath. Node of Ranvier, node of Ranvier, node of Ranvier. So those gaps are called nodes of Ranvier, and those yellow sheaths are called myelin sheaths. And they are produced by in the peripheral nervous system, they're produced by Schwann cells. Now, in the central nervous system, they're produced by something else, and we'll talk about that later on. All right, so again, Schwann cells in the peripheral nervous system produce these layers of myelin sheaths, and then we have those gaps called nodes of Ranvier. And you can see they kind of form these concentric circles, kind of like the tree rings if you chop down a tree. They can be a couple layers thick, or this one is actually many layers thick. 1, 2, 3, 4, 5, 6, 7, 8, 9, maybe 10 layers thick. I'm not sure. So this one is actually a pretty thick axon or a pretty thick myelin sheath. And it just wraps around the axon. All right, so myelinated axons. So we have, so sometimes we have axons that have that myelin sheath around them, and sometimes we have axons that don't have that myelination. So myelinated axons, they do have that myelin sheath, and when we look at them under a microscope, they appear white. Unmyelinated axons appear gray under a microscope. So if you have ever heard of white matter and gray matter, if you talk about the brain, that's what we're talking about. Gray matter is unmyelinated axons. White matter is myelinated axons. We've already said that myelinated axons are produced by Schwann cells in the peripheral nervous system. So Schwann cells are that neuroglia in the peripheral nervous system. In the central nervous system, this myelin is produced by these things called oligodendrocytes in the central nervous system. So oligodendrocytes would be neuroglia in the central nervous system. And we'll talk about the importance or the difference between Schwann cells and oligodendrocytes a little bit later on. All right, so myelinated axons in the central nervous system, white matter versus gray matter. And the importance of myelination, I mentioned earlier, is for conduction speed for electrical impulses. And I'll show you a picture of that here in a few slides. So unmyelinated axons, they don't have any myelin surrounding those axons. So no myelin, gray matter appears gray under the microscope. All right, so I mentioned that myelination is good for increasing the speed of conduction of that electrical impulse down the length of that axon. So, and it's dependent on the amount of myelination. So faster nerve conduction means that the nerve is myelinated. Slower nerve conduction means that the nerve is unmyelinated. And it says here, unmyelinated axons conduct impulses over the entire length, and I should say entire length of the axon. And like I said, I'll show you a picture of this here in the next slide. So remember that myelin is lipid soluble, so that water-soluble substances like ions can't cross that cell membrane, acts as an electrical insulator. And those ions can really only cross through those gaps. Remember, those gaps are called nodes of Ranvier. And when we have that myelin, myelinated axons, they transmit those impulses through this process called saltatory conduction. So those action potentials actually don't travel down that whole length of the axon. Remember up here I said that unmyelinated axons conduct impulses over the entire length of the axon, whereas in myelinated axons, we have this thing called saltatory conduction, where action potentials jump from node of Ranvier to node of Ranvier down the axon. So let me just write node of Ranvier to node of Ranvier down the axon. And I'll show you a picture of this so it's not as confusing. So saltatory conduction is actually much faster than on myelinated axons than on unmyelinated axons. All right, so here's our myelinated axon. And what we see with saltatory conduction is that the electrical impulse actually does not travel through or down the length of down the entire length of the axon. What will happen is that this electrical impulse will actually jump over this myelinated portion and will actually only, it will jump from node of Ranvier to node of Ranvier to node of Ranvier. So let's say that, and I'm just going to make up numbers here. It looks like that about what? 85% of this axon is myelinated, maybe we'll say. I don't know. So because 85% of this axon is myelinated, the impulse conduction time is 85% faster than it would be compared to an unmyelinated axon. Now, in an unmyelinated axon, let's pretend that we'll go down here to C. Let's pretend that this axon did not have any myelin on it. Then the impulse or that electrical impulse would actually have to travel down the entire length of the axon in order to get from point A to point B. So with myelin, that impulse actually jumps from node of Ranvier to node of Ranvier, increasing that impulse conduction time. So also, axon diameter affects conduction speed. So thicker axons transmit faster than thin axons. So if you want to say, if you want to make a blanket statement, you could say that thick axons with a lot of myelin are the fastest, produce the fastest conduction speeds. Thin axons with no myelin produce the slowest impulse conduction speeds. All right, multiple sclerosis, or MS. We actually abbreviate that as MS. Basically, this is a destruction of the myelin sheath in the central nervous system, so not peripheral, but central nervous system, by an immune response. Remember, an autoimmune disease is where the body attacks itself with its own antibodies, and it's an inflammatory response. And once the body attacks the myelin in the central nervous system, it leaves scars behind. So we call it multiple sclerosis because those scars are actually called sclerotic scars or scleroses. And those scars actually stops those neurons from conducting impulses. And it's typically on those efferent pathways, those motor neurons. And so if you don't have that motor control or that efferent pathway, your motor side is going to become weaker. So those muscles, those effectors are going to be diminished. They're going to stop contracting. They're going to undergo atrophy. Remember, atrophy is kind of like wasting syndrome. You're going to have some fatigue. You might have mood problems. blurred vision, weak, numb limbs. Remember also, effectors are not only muscles, but glands. So you could have possibly some dopamine, serotonin issues because those are neurotransmitters that are released by glands as well. And treatments involve medications that suppress the immune activity. So one of the treatments, actually, my sister has MS, and she's in remission right now. And she went through a study that involved bee stings. And so she was stung by, and she went through like a clinical trial phase where she would be stung by bees. And at one point, she was stung by 20 bees in a day. And that actually helped her body build up her own antibodies to attack the antibodies that were kind of killing off her myelin sheath. So the bees actually did some good for her. The bee stings did some good for her. All right, so looking at neurons, we actually see that we have three different kind of structures of neurons. Multipolar, and really, when we look at these neurons, we're looking at basically, we classify them basically how many processes are coming off of the cell body. In a multipolar neuron, and these are going to be most of the neurons that we see, 99% of the neurons. A multipolar neuron, we're going to have many extensions or many processes coming off of that cell body. Many dendrites, but only one axon. Remember, all neurons can only have one axon. So multipolar neurons can have many dendrites and one axon. So multipolar neurons, lots of dendrites. So here, 1, 2, 3, 4, 5, and then the one axon. So here, this one has 6 processes coming off of the cell body. In reality, we're talking about thousands of dendrites, but one axon. In a bipolar neuron, we only have 2 processes coming off of the cell body, one dendrite and one axon. We really don't see bipolar neurons that often, really only in our special senses, our eyes, ears, nose. We'll actually spend all of chapter 12 talking about our special senses, sight, sound, taste, touch, like that. So bipolar, 2 extensions only. So this is our bipolar neuron. We still have lots of dendrites, but we only have one main dendrite coming off of the cell body and one axon coming off of the cell body. Unipolar neuron. We also call them pseudo-unipolar. No one ever says pseudo-unipolar. So unipolar, one process extending off of that cell body. Those are going to be a lot of our sensory neurons. So a lot of our peripheral nervous system neurons, because our peripheral nervous system afferent neurons. Because remember, our peripheral nervous system afferent neurons detect changes inside of our body and from the outside world. So unipolar neurons. So unipolar neurons, we actually only have one process coming off of the cell body. So our dendrites come into the cell body, and then the information leaves by that same process. All right, so far we've talked about sensory neurons and motor neurons, or sensory afferent neurons. They carry impulses from the peripheral nervous system to the central nervous system, or the brain and spinal cord. And as we saw a few slides ago, most of those are going to be unipolar, some are going to be bipolar, and they're going to have the receptors or sensory receptors that are going to detect changes inside the body and from the outside world. Motor neurons or efferent neurons carry impulses from the central nervous system to those effectors, such as muscles or glands. Most of those are multipolar. And remember the difference between the somatic nervous system. Those are going to go to the skeletal muscles, our voluntary portion. Autonomic nervous system, our automatic portion. Involuntary, so smooth muscle, cardiac muscle, and our glands. But we also have interneurons. You're never going to see association neurons or internuncial neurons. Those actually are neurons that are isolated to the central nervous system. And they relay information from one part of the central nervous system to the other. They're multipolar neurons. Like I said, they're just found within the central nervous system. And I put this diagram here. So this will be the pathway of a nerve, of the pathway that you're going to have to memorize. So we have a receptor that is going to detect some sort of change. And then we have a sensory neuron or an afferent neuron. And then we have the interneuron or the integration center. So the interneuron, that's going to be within, and in this case, it's going to be the spinal cord. That's actually a cross section of the spinal cord. And then we have a motor neuron. And then we have the effector. In this case, it looks like a skeletal muscle. So you're going to have to memorize those 5 steps: receptor, afferent neuron, interneuron, efferent neuron, and effector. And we'll talk a lot more. I'll bring this up several times. All right, so again, peripheral nervous system, we have our sensory receptor, and that's going to go to the sensory neuron or the afferent neuron, the afferent pathway. And then it's going to go to the central nervous system. And first it's going to go to the interneuron in the spinal cord. And then we might have multiple interneurons. So let's say that we have, let's say that we're looking at our blood glucose level. So our sensory neuron in the inside of our body will have a chemoreceptor that is going to detect our blood glucose level. And it says that our blood glucose level is too high. Actually, it's going to say our blood glucose level is 247. And obviously, it doesn't come up with a number, but we'll say it comes up with a number. And then that sensory neuron will relay that information to here, the spinal cord. And then we have an interneuron that would go from the spinal cord up to the hypothalamus. And then, because remember, the hypothalamus does all of our negative feedback loops. And the hypothalamus would say, yep, our blood glucose level is way too high. We need to do something about it. So then we would have an interneuron that would relay that information back down from the hypothalamus back down to the spinal cord. And then we would have a motor neuron that goes from the spinal cord to, actually we'll do here, down to the pancreas to dump out some insulin. And then this whole process would get repeated until our blood glucose level is within a normal range. And then at that point, let's say our blood glucose level is, let's say 91. Our blood glucose level is 91. Sensory neuron would take that information to the spinal cord, to the hypothalamus. Our hypothalamus would say, yep, it's within normal range. And we would actually stop right there. Because it's in normal range, we actually would not have any motor output because we don't need any more insulin to lower our blood sugar because our blood sugar is within normal range. All right, so just a recap of difference between multipolar, bipolar, unipolar, sensory, interneuron, and motor neuron. All right, so neuroglia. Remember that neuroglia is that structural support for neurons. They provide that nourishment for neurons. They kind of clean up the excess ions, the neurotransmitters. They provide the oxygen and the nourishment for the neurons. They help form those synapses between neurons to muscles, neurons to glands. So a lot of structural support for neurons. And in the embryonic phases, they actually help guide neurons into the position that they're eventually going to be in. And they can help stimulate specialization of neurons. What type of neuron is it going to become? Is it going to become a bipolar neuron, a multipolar neuron, a unipolar neuron? So looking at the central nervous system, remember, we're actually going to have different neuroglia of the central nervous system versus the peripheral nervous system. So within the central nervous system, we're going to have these things called astrocytes. Astrocytes actually kind of look like stars. And they actually connect neurons to blood vessels. So just think about what that would do. Nutrients, growth factors, removing waste products, aid in metabolism, regulate ion concentrations. Look, it's linked up to the blood supply. And it also helps form scar tissue in that nervous system. We talked about multiple sclerosis. So that would probably be a part of the astrocytes, because remember, multiple sclerosis is a central nervous system disease. Oligodendrocytes, we actually mentioned this before when we talked about myelinization of those central nervous system axons. Also provides some structural support. Microglia, those are actually phagocytotic or phagocytic. They actually clean up some debris and some bacteria and also provide some structural support. Ependymal cells, they actually, we'll talk a little bit about this later on, they line the spinal cord. They line these things called ventricles of the brain. Ventricles in the brain, we have 4 ventricles of the brain that that's where we'll find our cerebral spinal fluid. And it's going to help regulate that composition of the cerebral spinal fluid. So we'll see some ciliated cuboidal columnar cells. Remember, ciliated, remember, has that these little hair-like structures on top of the cell. So you really don't have to know too much about what each of the neuroglia do, but you have to know what falls under the central nervous system and what falls under the peripheral nervous system. And we'll talk about the peripheral nervous system here in a second. So here, astrocytes, microglial cells, oligodendrocytes. Remember, oligodendrocytes produce that myelin in the central nervous system. Here's your regular neuron. Ependymal cells, remember that cilia in the ependymal cells. Ependymal cells associated with that cerebral spinal fluid. Astrocytes link up with those blood vessels, so they're going to be good for nourishment of the neuron. They're actually part of the blood-brain barrier. Remember, we're talking about the central nervous system. They have gap junctions, so they're going to be tightly packed together, allowing cell-to-cell communication. Like I said, provide nourishment to remove some of that bad gunk out of the neuron. All right, so here you can see the neuron here, another neuron here, but you can see a lot of the neuroglia. The neuroglia actually makes up the most of the composition of the central nervous system. Neurons don't make up a lot of the composition. All right, neuroglia of the peripheral nervous system. We already talked about Schwann cells. Schwann cells actually produce that myelin sheath on the axons in the peripheral nervous system. We already said that it speeds up the electrical impulse or the transmission of those electrical impulses through that saltatory conduction. Again, only found in the peripheral nervous system. We also have these things called satellite cells. And satellite cells actually support ganglia. We talked about ganglia, gave you the definition of ganglia, clusters of neuronal cell bodies within the peripheral nervous system. And their function is basically to nourish the neurons within the peripheral nervous system, help maintain that ionic concentration. So the ions, sodium, potassium, calcium, all of those ions that we find on that far left periodic table and the far right of the periodic table. So like I said, you have to know what falls under the central nervous system and the peripheral nervous system. All right, so organize this list by central or peripheral. Oligodendrocytes. Oligodendrocytes were central. Thermoreceptors. Where do we find thermoreceptors? You touch a hot stove. What part of the nervous system does that? You touch a hot stove with your hand. So that's your peripheral nervous system. Interneurons. Interneurons relay information within the central nervous system. Satellite cells. Satellite cells are part of the peripheral nervous system. The brain, that's your central nervous system. Brain and spinal cord is central nervous system. Motor neurons. So sensory neurons, motor neurons are part of the peripheral nervous system. All right, nerve regeneration. So Neurons do not go through mitosis. And if a neuron cell body is injured, typically that neuron is going to die off. Now, if you sever a nerve, let's say that you have your dendrite cell body and your axon. Let's say you sever a nerve. Sometimes that nerve can regenerate. If it's in the peripheral nervous system. If it's in the central nervous system, chances are it's not going to regenerate. But in the peripheral nervous system, if that axon is injured, it may regenerate. Now, that axon and that myelin sheath will degenerate, but those Schwann cells and that neurolemma will remain. Now, we didn't talk about the neurolemma, so let's kind of go all the way back to the beginning. Let's find that slide. So remember that we had that kind of outer coating, neurolemma right here. So your Schwann cells and that neurolemma will remain if that axon is injured or if that axon is severed. And that's what that is why nerve regeneration in the peripheral nervous system is possible. And the Schwann cells will provide, because those Schwann cells kind of stay there, that will act as like a guiding sheath for that regenerating axon, for that growing axon. Now, hopefully, that growing axon will recover its original function after being severed. If that growing axon reestablishes that former connection. But that function might be lost if that axon does not reestablish connection to where it once was. So here, let's say that you had your site of injury. That axon is going to degenerate. However, your Schwann cells act as that guiding sheath for that new axon to grow. And hopefully that connection will be reestablished. But there's no guarantee. And they typically say that it'll take up to 2 years. So, for example, June of 23, actually in May of 23, I herniated one of my discs in my back. And I had surgery in June of 23, and I had a lot of numbness and tingling going down my left leg, and my left foot was essentially numb. And so I know I had a lot of injury, nerve damage in my left leg as a result of my herniated disc. And in June of 23 is when I had my surgery. And after that, I immediately felt a lot better, but I still had numbness and tingling in kind of like my lateral 3 toes of my left foot. And So now I'm recording this video in May of 2025. And so I'm almost at that 2-year mark. And I still have a very, very minute amount of numbness in my lateral 3 toes of my left leg or my left foot. So I'm about 95% better. So I'm really close to that 2-year mark. So I would say most of the nerves that were injured did die off, and most of the nerves did reestablish connection, but I can tell that not all of them did reestablish connection. And I'm assuming that not all of them will, because I'm actually, let's see, 11 plus 10, 21, 22, 23. I'm about 23 days away from the 2-year mark. Anyway, so once I hit the 2-year mark, chances are if I still have the numbness and tingling, then I'll have it for, or the numbness, then I'll have it for the rest of my life. All right, so central nervous system, nerve regeneration. Highly unlikely. Regeneration is highly unlikely because the central nervous system, they don't have that neurolemma to act as the guiding sheath. Remember, in the peripheral nervous system, myelin is produced by Schwann cells. In the central nervous system, that myelin sheath is produced by oligodendrocytes. So therefore, the myelin does not have that neurolemma. And oligodendrocytes, they don't proliferate after an injury. So if you damage neurons in your central nervous system, regeneration is highly unlikely. All right, so now we're going to be switching topics to actually talk about the cell membrane potential and how this electrical impulse gets formed. So first I'll talk about the cell itself. If we look at the cell, and this blue line is my cell, we can see that there's a higher concentration of sodium outside of the cell compared to the inside. And that's why sodium is written bigger on the outside than inside. Calcium is bigger on the outside than inside, so that's why there's a higher concentration. So there's a higher higher concentration of calcium outside versus inside. Chloride, higher concentration outside versus inside. Potassium, higher concentration of potassium inside the cell versus outside the cell. So we can actually stick probes inside the cell and measure the voltage of a cell when it's at rest and when it's at various stages of when a cell is performing different functions. So at rest, a cell is electrically charged, or we say that it is polarized, so that we have kind of like a difference in electrical charge across that cell membrane. There's a charge difference, or it's polar. There's a polarity difference. There's a difference in its electrical activity from the inside of the cell to the outside of the cell. And we actually call that its membrane potential. And what we find is that the inside of the cell is actually negatively charged with respect to the outside of the cell. And that's just results from an unequal distribution of the ions from the inside of the cell to the outside of the cell. When the cell is at rest, when nothing's happening, we find that the charge is actually negative 70 millivolts inside the cell. Now, even though I wrote sodium, potassium, calcium chloride, there's a lot more. There's many more ions that are involved. There's many more molecules that are involved. Bicarb. There's glucose. There's a lot of things that are involved that make up this negative 70. But I didn't write them all. All right, so this this number is actually important in how we conduct impulses or firing that neuron and triggering contraction of skeletal muscle fibers, or triggering another neuron, or triggering a gland to release its contents. And these neurons are actually very excitable. We can actually change this negative 70, and if we change that negative 70 to a certain number, then we will kind of fire a neuron and create an action potential. So if you've heard the terms like depolarization, that's essentially our goal, is to depolarize this neuron. All right. So last chapter, I talked about the term threshold, and I said that we would talk about this next chapter. So we're going to be talking about that definition. All right, so we have this membrane potential. That's that charge inside of our cell. And right now I told you that it's negative 70 millivolts while it's at rest. And that's our potential to transport charges across that cell membrane. And that potential is because we have an unequal ion distribution of that cell membrane. And we've already said that we have a higher concentration of sodium outside the cell versus inside, and we have a higher concentration of potassium inside the cell than outside the cell. And from here on out, we're really not going to talk about calcium and chloride. We'll briefly mention chloride a little bit later on, but not much. Sodium and potassium are really our two key players in how a neuron becomes stimulated. All right, so it says gradients of sodium and potassium allow for excitability after a stimulus. Sodium and potassium follows rules of facilitated diffusion. Remember that facilitated diffusion, it's a passive process. It doesn't require energy. It doesn't require ATP. And it moves from an area of higher concentration to an area of lower concentration. So sodium's going to leak in here. Potassium's going to leak out. We follow our concentration gradient. But remember that the cell membrane is lipid soluble, so ions can't freely cross without some help, and that's why we have that facilitated diffusion. And I'll come back to this statement here that says resting membranes are more permeable to potassium than sodium. So it says that we have these ion-gated leak channels, and these ligand-gated channels allow for movements of these ions at certain times. Facilitated diffusion. Remember that we have these helper channels that allow for ions to move through the cell membrane, and that the ion would actually kind of come in here, and then the channel would open up, and then the ion would go through the cell membrane. That's facilitated fusion. We're going from an area of higher concentration to an area of lower concentration. So sodium would actually go this way, and potassium would actually go this way because we have a higher concentration of sodium outside the cell, so sodium would go inside the cell. Potassium would go the opposite way because we have a higher concentration of potassium inside the cell versus outside the cell. All right, so I said that we would come back to this statement of resting membranes are more permeable to potassium than to sodium. All right, so you don't have to worry about the math, but I just want to give you the concept behind why resting membranes are more permeable to sodium than to potassium. So I'm just going to draw my cell real quick here. Sodium versus sodium. And then we have potassium versus potassium. So higher concentration of sodium outside, higher concentration of potassium inside. And we can actually measure the concentrations of sodium extracellular fluid, intracellular fluid. We can actually measure the concentrations of potassium intracellular and extracellular. So if we look at potassium, so potassium intracellular is about 150 millimoles per millimoles per liter intracellular, and about 5 millimoles per liter extracellular fluid. So that ratio, 150 divided by 5, that's about 30-1. On sodium, intracellular is about 10, extracellular is about 140. So that ratio is 140 divided by 10 is about 14-1. And if you remember, when we talked about rates of diffusion or rates of facilitated diffusion, one of the factors that affects the rate of diffusion is the concentration difference or the concentration gradient. So potassium has a higher concentration gradient than sodium does. So potassium will diffuse more than sodium will diffuse. So that's why we have that statement that says resting membranes are more permeable to potassium than to sodium because we have a higher concentration gradient of potassium than we do to sodium. So you don't have to know the math behind that. You don't have to memorize the numbers, but I just wanted to explain why I put this statement in there. All right. Okay. All right, so hopefully this isn't going to be too confusing. I spent several hours on these slides. The first time I taught this, we actually spent about 2 days going over this. It was very confusing. And then kind of over last summer, I redid these slides. I spent about 10 hours on just these next probably probably 17 slides. So I'm hoping this is a lot less confusing. Last fall is when I updated the slides, and it actually went better last time I taught it. So hopefully it's going to be good. All right, so resting membrane potential. So the cell is at rest. Nothing is happening. Remember that I said we can stick probes in the cell and figure out what's going on. And that I said that our resting membrane potential is about negative 70 degrees, negative 70 millivolts. And what's happening here is that the cell is just doing its normal stuff. Things are moving in and out by diffusion if it's lipid soluble, and by facilitated diffusion if it's water soluble. Sodium's moving in and out, or sodium's moving in, potassium's moving out. If we go back up to this slide, chloride's moving in, calcium's moving in, potassium's moving out, sodium's moving in, bicarb, glucose, oxygen, carbon dioxide, everything is just moving in. So what we'll see is that this cell hovers around negative 70 millivolts. We might go to negative 69, negative 68, negative 71, negative 72. But essentially, on average, we're going to stay around negative 70. Resting membrane potential is negative 70. Now, I'm going to use these terms, ion-gated channels and ligand-gated channels. And that's to represent that ions are moving in and out, molecules are moving in and out. Usually by concentration gradients. Typically, all of these things are passive processes. They don't require energy in order to make this happen. All right, so at rest, sodium is moving in. At rest, potassium is moving out through these ion-gated channels or these ligand-gated channels. Now, sodium and potassium is going to be ion-gated channels, or we also call them leak channels. But if we have like a bigger molecule, glucose or bicarb, we might have to use these ligand-gated channels. Remember, I said that there were more molecules up here that I didn't draw. Typically, bigger molecules can't go through these ion-gated channels. They're going to have to go through these ligand-gated channels, where they have to have these receptors in order to work. All right, so neurons, we already know neurons are excitable cells. They respond to stimuli. So a stimulus essentially is anything that can change that resting membrane potential away from negative 70 millivolts. And it says in either direction. What we're going to talk about now is just the excitatory phase. But we can also have an inhibitory stimulus, which we'll talk about later. We have neurotransmitters like GABA that will inhibit neurons from being fired, which we'll talk about later. But for the next several slides, we're only going to be talking about excitatory stimuli. So neurons are going to detect that stimuli, and they're going to respond by changing that resting membrane potential. So we're going to have this local potential change. So we're going to have that change in membrane potential that occurs in that area of stimulation. And these local potential changes are graded. And that just means that the greater the stimulus, or the greater the intensity of that stimulus, the greater the potential change. So that just allows you to know if I'm whispering or if I'm shouting. It also allows you to determine if the water that splashes on your hand when you're cooking is lukewarm or if it's scalding hot. All right, so then we have this excitatory stimulus. The excitatory stimulus causes sodium to leak into the cell. And what happens is, remember, sodium is a positively charged ion. So what will happen is we're going to make this inside of the cell less negative or more positive. So you can see here that we're going to have that curve start to go up. So more positive, less negative. So right now we're still operating under these ion-gated leak channels, these ligand-gated channels. As we're pulling sodium in. Now, just some more definitions before we get to the next slide. We have these things called action potentials, which I've mentioned before. Basically, it's a sequence of electrical events once we excite the cell. We're going to change that cell membrane, first positive, then negative, to return to its resting potential. So eventually we're going to go really, really positive, and then really, really negative, and then eventually we're going to go back to its resting membrane potential. So let me do resting membrane potential. And it allows for potentials communication between cells. So when a neuron fires, it's going to actually trigger that action potential all the way down the length of the axon. And we call this the all or none response, or the all or none law. Once we trigger that action potential, it will send that signal all the way down the axon. Once we reach this magic number, it will trigger an action potential. There's no stopping it. All right. So right now, sodium is leaking into the cell, causing the cell to be more positive, or we could say less negative. Now, we mentioned last chapter this definition of threshold or threshold stimulus. We have it's basically an excitatory stimulus that causes changes in this membrane potential, and I'll just read the definition, where voltage-gated sodium channels open up and begin depolarizing the cell. All right, so we've reached threshold. So now we have two numbers to memorize. -70 is our resting membrane potential, and -55 is threshold. So we've reached threshold. Once we've reached that magic number of 55 millivolts, we have reached threshold. We will generate an action potential. There is no stopping this action potential from happening. We will depolarize the cell. Now, prior to this, we've been operating under ion-gated channels. We've been operating under these things called ligand-gated channels. At this point, ion-gated channels and ligand-gated channels are shut off. Because we are dealing with electrical impulses, now we switch to these things called voltage-gated channels. Voltage-gated channels deal with electrical impulses. So we've reached our threshold here of -55 millivolts. We shut off all of our leak channels. We shut off all of our ligand-gated channels. We've shut off essentially facilitated diffusion. Now we're going to switch to the electrical side of things. So now we're going to switch to these things called voltage-gated channels. So now we open up these things called voltage-gated sodium channels. So voltage-gated sodium channels open up, and sodium is going to rush into the cell. Which we'll talk about in the next slide. Now, here's this little statement: subthreshold stimulus does not result in action potential. So what that means is if I have, if I'm still in this phase right here and I don't reach that negative 55, let's say that I'm negative 62, I am sub-threshold. I haven't reached that magic number of negative 55. I'm not going to reach an action potential. And now we have this other statement that says graded stimuli can be added together to produce a threshold stimulus. We'll save that statement for later. All right, so we have now this phase called depolarization. So we were at threshold, we've reached threshold, and at threshold, remember that all ion gated and leak, or all of our leak channels and ligand gated channels are closed. We opened up our sodium voltage gated channels. And if we remember our cell, we had a higher concentration of sodium outside compared to inside. So now sodium rushes into the cell. We've opened up our sodium voltage gated channels. Sodium rushes into the cell. Sodium is a positive ion. So now we are making the cell more positive. So that's why we are going on an upswing here. We're making the cell more positive. We actually call this phase depolarization. So now both sides of the cell membrane are positive, because remember that this side of the cell membrane, the extracellular fluid, is positive, and now inside of the cell is positive. So now we go from negative 55 millivolts to about positive 30 millivolts. So now we have three numbers to memorize. Negative 70 is our resting membrane potential. Negative 55 is our threshold. And about positive 30 is going to be our peak. So here we've opened up our voltage-gated channels, our voltage-gated sodium channels, because remember, we've closed off all of our leak channels. We've closed off all of our ligand-gated channels. And we've opened up our voltage-gated sodium channels because now we are dealing with the electrical side of things. And voltage deals with electricity. Now, once we've reached that magic number of negative 55, we will generate an action potential. That action potential starts in that axon hillock. And that action potential will travel down the length of the axon. There's no stopping it. Remember that axon hillock, remember these are dendrites, cell body. That axon hillock is right here. It's where the axon comes off of the cell body. So this is where we start the axon, or that electrical impulse, and then it will travel down the length of the axon. All right, how does a neuron cell membrane become depolarized? sodium channels close and sodium can't diffuse into the cell. Calcium channels open and calcium diffuses out of the cell. Sodium channels open and sodium diffuses into the cell. Potassium channels open and potassium diffuses into the cell. So depolarization deals with sodium, so we can cross off those two. So our sodium channels open up and sodium diffuses into the cell. And if we actually want to be more correct, we should say sodium voltage gated channels open up and sodium diffuses into the cell. All right, so I just kind of made this question a little bit harder, but the answer is still the same. So sodium voltage gated channels open and sodium diffuses into the cell. Obviously, A and B are still wrong because we have shut off our ion gated channels. We operate under voltage gated channels, and for depolarization, we actually open up the sodium voltage gated channels. All right. Next question. And I forgot to say that, this is actually a positive feedback mechanism, because we actually, what we're doing here is we're making it worse, we're making it worse, we're making it worse, we're making it worse in hopes that we actually reset the cell to its resting membrane potential. So that's the definition of positive feedback mechanism. Remember, we make it worse and worse and worse in hopes that we reset it back to its original state. So that leads us to this question. A positive feedback loop occurs when the outcome of an event causes the event to continue occurring and amplifies its effect. Which of the following aspects of an action potential constitutes a positive feedback loop? Opening of a voltage-gated channel, voltage-gated sodium channels causes voltage-gated potassium channels to open and release potassium to the extracellular environment, slowly countering sodium influx. Initial flow of sodium ions into the neuron causes the opening of more voltage-gated sodium channels, rapidly causing more sodium to enter. The threshold is around -55. Sodium potassium pump exactly balances ion leaks by pumping 3 sodiums and 2 potassium ions across the cell membrane. So positive feedback means that we're making it worse, worse, and worse, and worse in hopes that we reset it. So here in A, we do open sodium channels, and eventually we will open up potassium voltage-gated channels. But here, this really isn't describing a positive feedback mechanism. This is actually just describing what's going on in depolarization and then repolarization, and we'll learn in repolarization that the word slowly is incorrect. So answer A is going to be out. Answer B, initial flow of sodium ions into the neuron causes the opening of more voltage-gated sodium channels, rapidly causing more sodium to enter. That's what we see in depolarization. We have sodium ions, more sodium, more sodium, more sodium, more sodium. So that's going to be our answer. That's a positive feedback loop. The threshold potential around -55, that's a blanket statement or a blank statement. That does not say anything about, that doesn't describe positive feedback mechanism. That's just a true statement. It doesn't say anything about positive feedback. Sodium potassium pump exactly balances ion link. That's a true statement, but we'll see later that this is actually going to be more negative feedback because it actually we'll see that it resets the cell in that something's too high, we need to lower it. Something's too low, we need to raise it. So this is actually a negative feedback loop right here, and we'll see that later on. All right, so we have this definition called propagation. And I said that once we create an action potential, it will go down the length of the axon. And that's just called propagation. How action potentials are related to impulses conducted down the length of the axon. All right, so now we have done depolarization, or we've done our resting membrane potential. We've had some sodium leak in. We've reached our threshold. We've closed off all of our ion-gated stuff. We've switched over to voltage-gated. We've opened up our voltage-gated sodium channels. Sodium has rushed into the cell. And now we've reached our peak at positive 30. So we're at the peak of our action potential. And like I said, it occurs around positive 30 millivolts. So we have to close off our sodium voltage-gated channels. And now we're going to open up our potassium voltage-gated channels. Remember, we're dealing with electricity, so we're dealing with voltage-gated channels, no ion-gated channels. Remember the ion-gated channels, ligand-gated channels, they are shut off. All right, so now we're looking at the downslope. This is called repolarization. Eventually, we're going to try and return to our resting membrane potential. I need to change that, resting membrane potential. And if we go back to our cell, we have a higher concentration of sodium on the outside versus inside. We have a higher concentration of potassium on the inside versus the outside. So basically, we're going to go from positive 30 back down to negative 70. And remember here, at the peak, we've closed off our sodium voltage-gated channels, and we've opened up our potassium voltage-gated channels. So our potassium voltage-gated channels are open, and potassium, which is a positive ion, rushes out of the cell. So because a positively charged ion rushes out of the cell, it's losing a positive charge, which is why we see an increase in the negative charge. We're losing positive charge from the inside of the cell. So that's why we see the downslope of that curve. But that's our resting membrane potential right there of negative 70. We actually don't stop at negative 70. We actually overshoot at the end of repolarization. We don't stop at negative 70. What we see is that we overshoot. We actually go to about negative 90 millivolts. We call this hyperpolarization. We actually become more negative than our resting membrane potential. So we actually go to about negative 90. So at some point during hyperpolarization, we have to shut off our potassium voltage gated channels. All right, so we have to, sometime during this period in hyperpolarization, we start to close our potassium voltage gated channels because essentially we're almost done with the electrical portion of our action potential. So we are going to turn on our potassium leak channels. So we'll say ion-gated leak channels. And then we're going to turn on our sodium-potassium pump, which we'll talk about in the next slide. All right, so for the sodium-potassium pump, remember that typically our cell has a higher concentration of sodium on the outside, and that we have a higher concentration of potassium on the inside. But because of our action potential, remember that sodium rushed into the cell, so we actually have a higher concentration of sodium on the outside now compared to the inside, and potassium rushed out of the cell, so we actually have a higher concentration of potassium on the outside of the cell versus the inside. So now we have to reset the cell with sodium potassium. So we have this thing called the sodium potassium pump that's going to return those ions to their original concentration. So it's going to balance those ions. It's an active process, so it uses ATP to actively transport these ions across that cell membrane. So what it does is it will pump 3 sodiums out, and it will pump 2 potassiums in to the cell. And once the sodium potassium pump does what it needs to do, our ending concentration will look like it should. Or like it originally did. All right, so once we get back to our resting membrane potential, remember that's our negative 70. We're going back to an inactive cell, negative 70 millivolts. Remember, our potassium voltage gated channels are shut off. We've turned off our sodium potassium pump because now we're going back to our cell is at rest. All of our ions and all of our other substances are moving in and out by regular diffusion, by facilitated diffusion. So our ion-gated channels, our leak channels, our ligand-gated channels, those are going to be turned on. We'll do this. All ion-gated channels and ligand-gated channels are turned on. Ligand-gated channels are active. All right, so here is a summary slide of kind of what happens. That nerve cell membrane, we have sodium, potassium moving by their regular concentration gradients, facilitated diffusion. That neuron receives a stimulation. We reach threshold. Sodium channels open up, those voltage gated channels open up. Sodium comes in, rushes inward. We go through depolarization. Potassium voltage gated channels open up. We go through, potassium leaves the cell. We repolarize that cell membrane. That action potential causes that electrical impulse to propagate or move down the length of the axon. So you might want to know this chart. So here's just kind of a summary slide of everything that we have talked about. At your resting membrane potential of negative 70 millivolts, we have our ion-gated and ligand-gated channels open up. Then at some point, sodium's going to leak in. Once we reach threshold at negative 55, all of our ion-gated channels and our sodium voltage-gated channels and our ligand-gated channels, all of our ion-gated channels, leak channels, ligand-gated channels, they're going to close, and we're going to switch to our voltage-gated channels. So here our sodium voltage-gated channels are going to open up. Sodium rushes into the cell, and we see that we go through this phase called depolarization. And then once we reach the peak, we're going to see that sodium voltage-gated channels start to close and our potassium voltage-gated channels start to open. Then our sodium voltage-gated channels are fully closed and our potassium voltage-gated channels are fully open. Potassium rushes out of the cell. Potassium voltage-gated channels start to close. We go into hyperpolarization. Our sodium potassium pump is turned on. 3 sodiums out, 2 potassiums in. Potassium leak channels, or those ion-gated channels, are open. Then we go back to our resting membrane potential of negative 70. All of our ion-gated or leak channels, our ligand-gated channels are open. All of our potassium voltage-gated channels are closed. Sodium potassium pump is turned off. And that's kind of it for how we generate an action potential or how we record an action potential. So I don't know if you remember last chapter, we had a statement. I'm going to pull it up real quick. Course information. I don't know if you can see this, but there is a statement last chapter that said acetylcholine causes changes in the membrane permeability to sodium influx and potassium efflux, which generates a muscle impulse or action potential. It was on slide 21 of chapter 9, and I said we would save that statement for next chapter. So acetylcholine causes changes in membrane permeability to sodium rushing into the cell and potassium rushing out of the cell, which generates that muscle impulse or that action potential. And I said that we would save that statement for next chapter. So that's what that means. Sodium rushing into the cell and potassium rushing out of the cell. All right, so question. What is the correct sequence of events that occurs along the action after the membrane reaches threshold? All right, so the membrane depolarizes, sodium channels open and sodium ions diffuse inward. The membrane repolarizes, potassium channels open and potassium ions diffuse outward. All right, so we know that sodium goes before potassium, and we know that depolarization happens before repolarization. So we know that 1 and 2 comes before 3 and 4. So sodium channels open and sodium ions diffuse inward, and then the membrane depolarizes. Then potassium channels open and potassium ions diffuse outward, and then the membrane repolarizes. So that would be your answer. All right, a neuron is exposed to a drug that completely blocks voltage-gated potassium channels, but has no effect on non-gated channels or the sodium-potassium pump. Which of the following describes the effect on action potential generation? So first, I'll say non-gated channels. So it completely blocks voltage-gated channels. So these non-gated channels, those are going to be your ion-gated channels, your leak channels, your ligand-gated channels. All right, so resting membrane potential would not be maintained as potassium could not leak out of the neuron. So a drug that completely blocks voltage-gated potassium channels. So basically, we have reached threshold and we have depolarized the membrane. So A would be out because we've already done resting membrane potential, we've done threshold, and we've done depolarization. All right, repolarization would still occur, but at an ineffectively slow rate. We'll come back to that. Repolarization will never occur after threshold is crossed, and sodium will continue to flow into the neuron. Oops. Well, no, because it says that it blocks voltage-gated potassium channels. Our voltage-gated sodium channels still work. So our sodium voltage-gated channels would be closed. So sodium would not continue to flow into the neuron. So that one would be out. Threshold will never be crossed. Well, no, because we've already reached threshold. Because remember, potassium is repolarization. We've already reached threshold. All right, so B is going to be our answer. repolarization will still occur, but at an ineffectively slow rate. All right, so let me get rid of this. So if you remember, our action potential kind of looks something like this. Now, because our voltage-gated potassium channels are blocked, our voltage-gated potassium channels are blocked, but our ion-gated channels work fine. Our sodium-potassium pump works fine. What we would see is that repolarization would still occur, but at an ineffectively slow rate. So our graph would actually look something like this. We wouldn't see that sharp decline in our repolarization phase because the voltage-gated potassium channels are blocked. So in that case, the ion-gated channels, our leak channels, and our ligand-gated channels would be the backup, and our sodium-potassium pump would be the backup as well. However, they don't work as fast as the voltage-gated potassium channels. So repolarization would still occur, but it would be much slower. All right, refractory period. So there is a time period in which during an action potential that you cannot generate another action potential. That is called an absolute refractory period. Absolute refractory period is the time in which you cannot generate another action potential. And typically this is seen when your voltage-gated sodium channels are still open. So that's going to be during depolarization, during that upswing on the curve. We also have another type of refractory period called a relative refractory period. And this relative refractory period is basically you can generate another action potential, but it's going to take a lot of work in order to create that action potential. And typically, that action, this relative refractory action potential is going to be seen when you're in that hyperpolarization phase. So right here. So we're hovering around that negative 90 millivolts. And remember, we have to get up to negative 55. So repolarization is not complete. That membrane is not reestablished. We're not back at resting membrane potential. So it's going to take a lot of work in order to get back up to that negative 55. But we can do it if we have enough positive ions that are rushing into the cell. All right, some factors that affect impulse conduction. Typically, remember that we're talking about sodium and potassium. This slide really only talks about potassium, but we do have disorders of sodium as well. So if we have increases of potassium in that extracellular fluid, so remember potassium has a higher concentration inside the cell versus, actually, I should have drawn my K smaller, inside the cell versus outside the cell. So if we have a higher concentration of potassium in the extracellular fluid, that's going to decrease that concentration gradient. So not much potassium actually leaves the cell. So that threshold is going to be reached with a lower intensity. So your neurons are actually more excitable. And that can lead to convulsions. Decrease in concentration of potassium in the extracellular fluid. Neurons are hyperpolarized. So if you look at that's a bad picture. So your neurons kind of live down here in that hyperpolarization phase. It's going to take more energy to get to that negative 55. It's going to take more positive ions to get to that negative 55. Action potentials that typically go to threshold are now not getting to threshold. So that could lead to paralysis or not generating action potentials. Decrease in permeability to potassium. Those are your anesthetic drugs. Impulses don't reach the brain. Perception of touch, pain. Potassium is one of those ions. For those of you going into nursing or the medical field, you really, really have to pay attention to your lab values. Potassium, whether you're hyperkalemic or hypokalemic, you have too much potassium or too little potassium, can really, really affect your heart. It can produce, whether you're too much potassium or too little potassium, it can really affect the electrical activity of the heart. So normal potassium, 3.5 to 5.3. Really, really monitor your potassium levels. All right, so we've already talked about the synapse. When we talked about the myoneural junction or the neuromuscular junction. Nerves can communicate with each other at a synapse. When we're talking about two nerves, we have what's called a presynaptic neuron and a postsynaptic neuron. And when we draw neurons, we actually draw them kind of like this. That's our cell body, and that's our axon. Then we have another cell body and another axon. We really don't draw the dendrites. So this would be our presynaptic neuron, and this would be our postsynaptic neuron. So our presynaptic neuron is what would release the neurotransmitter, and that postsynaptic neuron receives that impulse. Synaptic transmission. Remember that it is a one-way street only. One-way street only. Dendrite, cell body, axon. Dendrite, cell body, axon. And we've already looked at the synapse before, last chapter. We're not going to look at it again right now. We've already looked at that synaptic cleft. Remember our vesicles. We have to have calcium in order for those vesicles to fuse to the cell membrane to release the neurotransmitter. Remember, for skeletal muscle contraction, that neurotransmitter was acetylcholine. All right, so actions that lead to neurotransmitter release. That action potential actually goes, remember, all the way down that axon over that synaptic knob. This is that synaptic knob right there. Calcium has to be involved. Calcium diffuses into that synaptic knob. Then those synaptic vesicles, those vesicles that are in that synapse, fuse to that membrane of that synaptic knob. Then that neurotransmitter gets released by exocytosis into that synaptic cleft. Remember, exocytosis is an active process that requires ATP to do so. And then that vesicle becomes part of that cell membrane. So this vesicle, once it fuses to that cell membrane, that vesicle becomes part of that synaptic knob's cell membrane. And then we have a statement here that added membrane provides material for endocytotic vesicles. So we talked last chapter about acetylcholinesterase. So once acetylcholine gets dumped out in the synaptic cleft, acetylcholinesterase will break down this acetylcholine. However, some neurotransmitters don't get broken down by a chemical or a compound. Some neurotransmitters get taken back up into that synaptic knob. So they would actually get repackaged into these vesicles in that synaptic knob. So that's what this statement is. If you remember, the opposite of exocytosis is endocytosis. So that's this statement. That added membrane provides material for endocytosis, for packaging those vesicles to be reused, so that this neurotransmitter would actually go back up into the extra neurotransmitter would get repackaged into that into vesicles into the synaptic knob. All right, so we kind of covered some of this on the last slide. And the slide, a couple slides before. So the transmission of a nerve impulse from one neuron to another. We drew the neuron with the presynaptic neuron to the postsynaptic neuron. So that impulse will stimulate that presynaptic neuron or that axon, which causes that neurotransmitter to be released in that synaptic cleft. And then the dendrites of that postsynaptic neuron will pick up the neurotransmitter that was released by that presynaptic neuron. Now, the effects of that neuron, the neurotransmitter, will vary. Like I said before, we have about 100 different neurotransmitters. We talked about acetylcholine already, and that will contract. That's an excitatory neurotransmitter for skeletal muscles. So some neurotransmitter will open up ion channels, and others will close ion channels. So some neurotransmitters are considered excitatory. And some neurotransmitters are considered inhibitory. So excitatory neurotransmitters increase the permeability to sodium. So if you go back to our our action potential graph, increasing the permeability to sodium would actually move it this way. It brings that membrane closer to threshold and increases the likelihood of generating impulses. Inhibitory neurotransmitters move that membrane farther away from threshold. So our action potential would actually go this way. We would actually go from like -70 down to, -90, -100. So it decreases the likelihood of generating impulses. So in our action potential example that we went through, all we looked at was the excitatory portion of a neurotransmitter. We did not look at the inhibitory portion of a neurotransmitter because that would never reach threshold. All right, a person is exposed to a drug that prevents vesicles from forming in neurons. What effect will this have on nerve functions? Neurons won't be released into the synapse. Action potentials will not be carried along the axon. Voltage-gated potassium channels will be blocked, slowing depolarization, and slowing nerve impulse. Neurons will be hyperexcitable. Well, what do vesicles store? Vesicles store neurotransmitters. If you have a drug that prevents vesicles from forming, vesicles store neurotransmitters. Well, if vesicles can't form, then neurotransmitters can't be released in the synapse. So that's our answer. Now, action potentials will still be created because action potentials travel down the length of the axon, and then the neurotransmitter will be released. Voltage-gated potassium channels will be blocked, slowing depolarization. No, because we've already created the action potential. Neurons will be hyperexcitable. No. All right, so far we've talked about kind of one neuron or a presynaptic and a postsynaptic neuron kind of in isolation. In reality, neurons, it's thousands of neurons that are communicating with thousands of neurons. We have these terms convergence and divergence. And in textbook definition, convergence is 10 or more presynaptic neurons that stimulate or inhibit 5 or fewer postsynaptic neurons. So here's just a picture of convergence, where here it's just showing 2 neurons that are stimulating 1 neuron. But remember, the textbook definition is 10 neurons that stimulate, or 10 or more presynaptic neurons that are conducting impulses to 5 or fewer postsynaptic neurons. Axons from the neurons that actually contact the same neuron. So you can see that multiple axons from different neurons are actually coming in contact with the same postsynaptic neuron. And that postsynaptic neuron will collect, process, and respond to all of that information coming in from all of those different neurons. And it says here, it makes it possible for a neuron to sum impulses from different sources. And we'll talk about that statement here in a couple different, and in about 2 slides. Divergence is the opposite of convergence. One neuron sends impulses to several different neurons, stimulating many neurons. So this one is divergence. You have one neuron stimulating here 2 neurons or many different neurons. So one axon branches off into collaterals, stimulating here 2 neurons. Here it will amplify the impulse. We actually saw this in our skeletal muscle example. We call that recruitment. And so this is how we contract the whole bicep muscle as opposed to just a single myofiber. So that several motor units are involved as opposed to just a single muscle fiber. And in reality, here we're doing 2 to 1 or 1 to 2. In reality, each neuron, we're talking about thousands of axons. So we could be talking about thousands of axons. We could be talking 10,000 axons to 1,000 axons, or 1,000 axons to 10,000 axons. All right, so excitatory postsynaptic potentials, EPSPs, and inhibitory postsynaptic potentials, IPSPs. So this is excitatory. If you remember, on our depolarization side, that's sodium, and our repolarization side is potassium. So if you think about excitatory or a depolarization, you can think of sodium. If you think about what is inhibiting or repolarizing the cell, that's going to be potassium. So sodium, you can think about exciting the cell. What's going to generate an action potential? Potassium would be making the cell less negative. Remember, potassium is a positively charged ion, and when potassium leaves the cell, it makes the cell less negative, so we see that downslope. So potassium is going to be an inhibitory ion. So think when sodium is a positively charged ion, when sodium rushes in the cell, it makes it more positive. So that's why we see the upswing on the sodium side or the depolarization side. So you can think of sodium as an excitatory ion and potassium as an inhibitory ion. All right, so excitatory postsynaptic potentials open those sodium channels and makes that membrane less negative. And we'll see that depolarization of the membrane. So sodium is going to rush in, generates that action potential, or that action potential has become more likely, and it lasts about 15 milliseconds. Inhibitory, think of potassium. So we're going to open up those potassium channels, and I'll get to chloride here in a second. Remember, potassium is inhibitory, and it can hyperpolarize as potassium leaves the axon. So potassium positively charged leaves that cell, makes that cell more negative, makes that membrane more negative. Therefore, the chance of the action potential happening will be less likely. Now, I said I'd come back to chloride. Chloride is that negatively charged ion. So, negatively charged ion, we have a higher concentration of chloride outside versus inside. If we follow our rules of facilitated diffusion, chloride coming into the cell would make that cell more negative. So we would go this way. So chloride coming into the cell makes it more negative. So it's actually an inhibitory ion. Makes it more negative. Inhibitory action potential becomes less likely. All right. Now, I said that we have convergence and divergence, and we have, thousands of different neurons that are communicating with thousands of different neurons. So let's just talk about let's look at the 1,000 different neurons compared to that one neuron. Now, what happens with that one neuron that's receiving information from the thousands of different neurons? We're going to look at, we have to do this process called summation. We have neurons that are either excitatory or inhibitory. And you can see here in this picture that, there's many different neurons connected or synapsing with this one neuron right here. Some are going to be excitatory, some are going to be inhibitory. But what is the net result of the neuron that it's stimulating? Will this neuron be an excitatory neuron, or will it be an inhibitory neuron? Will it generate an action potential at that axon hillock, or will it be an inhibitory neuron? What we have to do is look at those EPSPs and those ISPs, and we have to do this summation process. What we'll do is we're not going to do it. The body does it automatically, but what we could do is we could stick a probe in all of those thousands of neurons that are stimulating that one neuron and look at what the millivolts are of all of those thousands of neurons and determine what the millivolts are of all of those thousands of neurons. And then we can add them up and then do an average of those. And whatever the average is, that's going to be that's going to determine whether or not it's that's going to determine whether or not that post-synaptic neuron is excitatory or inhibitory. So if we add it up and that net summation is excitatory, then we'll probably create an action potential. If that net summation is less than -55, then we're not going to generate an action potential. So the action potential is going to be generated if those excitatories outweigh the inhibitories. All right, so, we look at the axon terminals from several different neurons that synapse into one single neuron. So basically, we're looking at a convergence a converging neuron. Some of these presynaptic neurons send excitatory signals while others send inhibitory signals. Okay, we already know that. Over the course of these effects, these signals, the postsynaptic membrane is negative 90 millivolts. So we've stuck in probes in all of those several different neurons, and we said that the net result is negative 90 millivolts. What's the overall effect of that postsynaptic neuron? The postsynaptic neuron is stabilized and remains at its resting membrane potential. Postsynaptic neuron is inhibited from generating action potentials. Postsynaptic neuron is excited, but not enough to create threshold or cross threshold. Postsynaptic neuron is excited enough to easily cross threshold and generate an action potential. All right, so basically this question is asking, where does negative 90 millivolts fall on this scale, on this drawing? So, is it at resting membrane potential? Resting membrane potential is negative 70. No. Is it inhibited from generating action potentials? Well, what's our magic number? Our magic number is negative 55. Negative 90 is much less than negative 55. So this is actually an inhibitory neuron. So that's going to be our answer. The postsynaptic neuron is excited, but not enough to cross threshold. So in this case, we'd have to be between negative 70 and negative 55. Negative 90 does not fall between there. The postsynaptic neuron is excited enough to easily cross threshold and generate an action potential. So that would have to be somewhere greater than negative 55. Negative 90 is much less, so B is going to be our answer. All right, neuronal pools. Neuronal pools basically, remember that I told you that ganglia is a cluster of neuronal cell bodies that lie outside of the central nervous system. Neuronal pools basically are groups of interneurons that make synaptic connections with each other that are inside the central nervous system. So they're almost like ganglia inside the central nervous system. There are a bunch of cell bodies within the central nervous system, and they're going to they kind of work together to perform a common function. They receive input from other neurons, and they also generate output for other neurons. We're not really going to talk much about neuronal pools, so just really just know the definition of neuronal pools, that being groups of interneurons within the central nervous system. And that should be the key for interneurons, because interneurons really are only within the central nervous system. All right, facilitation. Facilitation is basically impulses on a presynaptic neuron. And it's usually due to that permeability of sodium or that stimulus of sodium. Remember that our resting membrane is negative 70 and we're trying to get to negative 55. And we dump in a little bit of sodium rushes in, a little bit of sodium rushes in, a little bit of sodium rushes in, but not enough to reach that threshold of negative 55 to generate an action potential. So we say we're sub-threshold. So we're not there yet, but we're moving in the right direction to create an action potential, but we're just not there yet. So we're increasing the likelihood that that postsynaptic cell will reach threshold, but as of now, we're not there yet. We haven't reached that magic number of negative 55, but we're trying to get there. So the example here is we're going from negative 70 to negative 60. We still haven't reached negative 55 yet. All right, so for neurotransmitters, I've said before, we have about 100 different neurotransmitters. We already know acetylcholine is the neurotransmitter for skeletal muscle contraction. Neurotransmitters can be monoamines. They could be modified amino acids, dopamine, serotonin, epinephrine. Those are all modified amino acids. Peptides. If you look at all of these, a lot of these are proteins. Amino acids are the building blocks of proteins. We all know that by now. Neurotransmitters are produced in the rough endoplasmic reticulum. Remember what the rough ER does. Protein synthesis. All right, when an action potential passes over that surface of the synaptic knob, remember, calcium channels are open. We have that synaptic knob, and calcium has to be present in order for those neurotransmitters to be released. So presynaptic neurotransmitters released by exocytosis. We've mentioned that, or I mentioned that several times now. Exocytosis, it's an active process. It requires ATP. All right, vesicle trafficking. I've mentioned this before. Vesicles fuse with the cell membrane, dump out their parts, dump out their gunk by exocytosis. And that synaptic vesicle becomes part of the cell membrane. And then sometimes some neurotransmitters get taken back up into that synaptic knob by endocytosis and forms new vesicles. That's called reuptake, where that neurotransmitter goes from that synaptic cleft back up into that synaptic neuron. Remember, acetylcholinesterase or acetylcholine does not work that way. Acetylcholine gets broken down by acetylcholinesterase. Some neurotransmitters that are involved in reuptake is dopamine, norepinephrine, GABA, serotonin. Dopamine is good for reward pathways, motivation, norepinephrine, alertness, arousal. Norepinephrine is very closely related to epinephrine. That's adrenaline. GABA is an inhibitory neurotransmitter. It helps you calm you down. Serotonin, also mood regulator. All right, just some neurotransmitters that you're going to have to know. Acetylcholine, we already know, skeletal muscle. Dopamine. Dopamine creates a sense of well-being. Nitric oxide. In the peripheral nervous system, it causes vasodilation. So the interesting thing about nitric oxide is vasodilation, expansion of the blood vessels. So there's a medication out there now that it was formed or formulated for a blood pressure medication to lower blood pressure, and it caused vasodilation. When it went through human trials, they noticed a lot of side effects. And one of those, and because of those side effects, they actually discontinued the trials of blood pressure medication, and now they started marketing it and reformulated it as an alternative medication. So if you have ever heard of Viagra, that's what I'm talking about. Viagra affects nitric oxide, and Viagra was once going through clinical trials as a blood pressure medication to vasodilate and lower your blood pressure. However, once it started going through human trials, they noticed all of these side effects in men, and now it's marketed as well what you know it as. All right, so more or other disorders that are associated with neurotransmitter imbalance. Clinical depression is a deficiency in serotonin. You're going to have to know what disorders are treated by increasing the action of serotonin. So you want to keep serotonin in that synapse longer. Hypersomnia, excess serotonin. Insomnia, deficient serotonin. So with your deficiency of serotonin, we have these things called selective SSRI, selective serotonin reuptake inhibitor. So if you break it down, selective serotonin reuptake inhibitor. So selective serotonin reuptake inhibitor would actually, the SSRI, or the selective serotonin reuptake would, serotonin gets taken back up from that synaptic cleft, back up into the synapse, as we saw, here. And we have these SSRI medications, selective serotonin reuptake inhibitors, that would actually try and keep serotonin in that synapse longer. All right, medications or drugs that affect or alter neurotransmitter levels. Monoamine oxidase inhibitors is the one that you're going to have to know. It affects norepinephrine. So it actually blocks the degradation of norepinephrine in that presynaptic neuron. Nicotine affects acetylcholine. We'll talk about that next chapter. Cocaine affects dopamine. It actually blocks the reuptake of dopamine. So dopamine stays in that synaptic cleft longer, which is why you get that sense of euphoria. Same thing with nicotine and dopamine, that you get that sense of pleasure. Let's see, antidepressants affect both norepinephrine and serotonin. It blocks that reuptake. I talked about SSRIs a minute ago. We also have this SNRI, selective serotonin norepinephrine, or selective norepinephrine reuptake inhibitor. We have an SSNRI, selective serotonin and norepinephrine reuptake inhibitor. Or it's also called a dual reuptake inhibitor. All right, neuropeptides. Neuropeptides are usually synthesized in the brain or spinal cord. And some neuropeptides can act as neurotransmitters, and others can act as these things called neuromodulators. Neuromodulators basically alter a neuron's response to a neurotransmitter, or they can block the release of a neurotransmitter. And some examples, we have these things in our body called enkephalins. And what enkephalins do is they're like our natural, or they help relieve pain sensations in our body. We also have these things called endorphins or beta endorphins. They also help relieve pain sensations, more long-lasting pain sensations. We also have this compound called substance P that's found in neurons that conduct pain impulses. So substance P conducts pain impulses, and both enkephalins and endorphins both inhibit the release of substance P. So enkephalins and endorphins are kind of like our body's natural Tylenol or Advil. Opiates. So opiate medications or drugs, morphine, heroin, codeine, opium, very potent painkillers that are derived from this poppy plant. And humans actually produce these opiates called endorphins. And we can actually relieve our pain, our own pain, because we have receptors for endorphins, or we can take opiates because we have those endorphin receptors. We can also relieve pain through acupuncture because of those. We can take endogenous opiates, analgesia, childbirth, feeling of well-being during exercise, that natural endogenous or that natural endorphin release. Opiate drugs, very useful in relief from severe pain. However, it is very addictive. A lot of physicians won't write a prescription for more than 10 days. Some are going now to 4 days, especially in Robeson County. A lot of drug addiction in Robeson County. Withdrawal symptoms. After someone who is addicted stops taking the drug or medication, because that person is now short of their own endogenous endorphins, and they can actually feel what's called hyperalgia, that their pain is now at a heightened perception because they don't have that natural or that endogenous endorphin release. All right, drug addiction. Those drugs actually alter the release of neurotransmitters on the postsynaptic neuron. And we're not going to really get into the rest of this slide. All right, so a certain drug decreases the membrane permeability of nerve fibers to sodium. What would be a possible effect? An increase in the amount of nerve impulses through an affected area, an increased sensitivity to pain, a decrease in pain, or an increase in pain. So think about our curve and what happens And where is sodium? Sodium is on this side. And remember, we talked about excitatory versus inhibitory. So sodium is excitatory. All right, so if sodium is excitatory, we would not see a decrease in pain. Or I'm sorry. Let me back up. It decreases the membrane permeability of nerve fibers to sodium. So C would actually be our answer. We would see a decrease in pain. All of the other answers would be if we had an increase in membrane permeability to sodium. So if you decrease the membrane's permeability to sodium, you're less likely to reach threshold. Therefore, you won't generate an action potential. Answers A, B, and D all have to do with generating an action potential or sensation, which is felt because you generated an action potential. All right, so that is all I have for this chapter. If you have any questions, please let me know. Otherwise, I will see you for next chapter. All right, take care.